New techniques in biochemistry. The field of biochemistry has greatly advanced since its beginning in the mid-1900s. New techniques have been developed that allow for a more detailed analysis and understanding of many biological molecules and processes in a living cell. These techniques offer a wide range of applications to all scientific fields. They are mainly used in biochemistry to study the structure and properties of biological molecules, to understand the molecular movement of these molecules within larger systems, and to determine how these molecules interact with one another. There are many tools by which biochemists can view the structure of molecules. One example is the electron microscope that allows biochemical researchers to directly view the details of a microscopic specimen. A specimen may be a cell or large biological molecule such as a protein. An electron microscope can magnify an object up to 2 million times its original size by passing a beam of electrons through the specimen, and then focusing those electrons through a lens. The electrons form an image on a photographic plate. The electron microscope not only has tremendous magnification power, but it also has an extremely high resolution. These properties allow scientists to view parts of cells that are otherwise invisible to the naked eye. X-ray crystallography is another method biochemists use to study the structure and makeup of complex biochemical substances like enzymes and nucleic acids. X-ray crystallography shines a beam of X-rays through a molecule. The atoms in the molecule act as tiny mirrors. They spread out the X-rays into regular patterns. These patterns are captured on a photographic plate. By studying the patterns, scientists can determine how the atoms of various intercellular molecules are arranged. Still another technique for obtaining information on molecular structure is nuclear magnetic resonance spectroscopy. This technique is more commonly known as NMR spectroscopy. NMR spectroscopy places biological molecules in a magnetic field. It then measures the rate at which the protons in these molecules vibrate. The protons of different chemical elements in a molecule will vibrate at different rates. Scientists can thus determine the types of atoms present in organic molecules. Biochemists also wish to know how molecules move in cells and throughout the bodies of living organisms. The predominant method for tracing organic substances in a biological system is referred to as radioisotopic labeling. This technique involves labeling a substance with radioactive elements called radionuclides. Hence, a substance is marked in order to track its passage through a system. It is labeled by adding radionuclides to its chemical composition. The radionuclides emit radiation that can be detected as they move through a system. The precise movement of a substance containing radionuclides can be easily traced by measuring radionuclide emissions. The biological processes in living systems do not result only from the types of molecules present and the movement of these molecules throughout the body. They also happen because of interactions between molecules. Some molecules will chemically react with one another to create products. The products are used by cells for activities like energy storage or cell division. Chromatography is a method that allows biochemists to separate and identify the products of chemical reactions. Chromatographic techniques involve a process called absorption. A chemical mixture is passed through a solid or liquid material. This material absorbs the substances in the mixture. Different substances, however, will be absorbed at different rates. By allowing the mixture to pass through the absorbent at a specific rate, only some substances are absorbed. This separates the mixture into its various parts. Because reactions between molecules follow the laws of chemistry and physics, biochemists can mathematically predict how atoms and molecules of substances will interact with each other in a given system. Most biological systems are made of a large number of particle interactions. This makes them extremely difficult to study using only analytical methods. Therefore, in addition to laboratory experiments, biochemists use a technique called molecular dynamics. Molecular dynamics uses a computer to act out the interactions between multiple atoms and molecules. The program is based on known chemical and physical laws. Combined with other new techniques in biochemistry, molecular dynamics offers a broader understanding of biological systems and life itself.